Welcome to Module 1 of our course on DNA typing, proteomics and beyond, recombinant DNA technology. In this first part, we will begin with an in-depth exploration of DNA typing, the scientific principles, molecular foundations, and analytical methods used to distinguish one individual's genetic makeup from another's. We will then move into the foundations of recombinant DNA technology, setting the stage for the more advanced proteomics and applied topics covered in Module 2. Throughout this lecture, the content will flow in a way that both builds conceptual understanding and highlights real-world applications in medicine, forensics, biotechnology, and research. 1. Overview of DNA typing. DNA typing, sometimes referred to as DNA profiling or genetic fingerprinting, is the process of analyzing specific genetic markers to identify individuals or establish biological relationships. The key to its power lies in the variation found within human DNA. While all humans share more than 99.9% of their DNA sequence, the remaining fraction contains unique patterns that can serve as a biological signature. These variations are called DNA polymorphisms. 2. DNA polymorphisms, the basis of DNA typing. Definition and significance. A DNA polymorphism is a variation in the nucleotide sequence at a specific position in the genome that occurs in a significant portion of the population. For a change to be considered a polymorphism, it generally must be present in at least 1% of individuals. Polymorphisms occur in both coding and non-coding regions of DNA. In DNA typing, the non-coding, highly variable regions are often most valuable because they do not undergo the selective pressure that limits variability in essential genes. Types of polymorphisms. Single nucleotide polymorphisms, SNPs, variations at a single nucleotide position. Variable number tandem repeats, VNTRs, longer sequences of DNA that repeat in tandem and vary in length between individuals. Short tandem repeats, STRs, shorter sequences of 2 to 6 base pairs repeated in tandem. Insertion/deletion polymorphisms, indels, small sequences present or absent in different individuals. These variations form the molecular foundation for all DNA typing methods. 3. Minisatellite analysis. Minisatellites are VNTRs with repeat units typically 10 to 60 base pairs in length. They are dispersed throughout the genome, often in non-coding regions, and exhibit high variability between individuals. Historically, the first widely used DNA profiling method, developed by Sir Alec Jeffreys in the mid-1980s, relied on probing these minisatellite regions. Principle. Restriction enzymes are used to cut DNA into fragments, which are then separated by gel electrophoresis and transferred to a membrane in a process called Southern blotting. Radioactively labeled probes that bind to the minisatellite sequences reveal a binding pattern unique to an individual. While this method has largely been replaced by PCR-based approaches due to its complexity and time requirements, it set the stage for modern forensic DNA analysis. 4. Polymerase chain reaction, PCR-based analysis. Overview. PCR revolutionized DNA typing by allowing scientists to amplify specific DNA regions from very small or degraded samples. The process involves repeated cycles of denaturation, primer annealing, and extension using a thermostable DNA polymerase. Advantages. Extremely sensitive, works with nanogram amounts of DNA. Rapid turnaround time. Compatible with highly degraded DNA, such as that from old forensic samples. PCR is now the backbone of most DNA typing approaches, including STR and mitochondrial DNA analysis. 5. Short tandem repeat STR analysis. STRs are the current gold standard for forensic DNA profiling. They are highly polymorphic, relatively easy to amplify by PCR, and distributed throughout the genome. Procedure. DNA is extracted from the sample. STR loci are amplified by PCR using fluorescently labeled primers. Products are separated by capillary electrophoresis. The number of repeat units is determined for each STR locus, generating a numeric profile. By combining results from multiple STR loci, typically 13 to 20 in standard forensic panels, the probability of two unrelated individuals sharing the same profile becomes astronomically low. 6. Mitochondrial DNA, mtDNA, analysis. Why mtDNA? Present in hundreds to thousands of copies per cell. Circular, double-stranded genome inherited maternally. Useful for analyzing samples where nuclear DNA is degraded or absent, e.g., hair shafts, bones, ancient remains. Limitations. Lower discriminatory power compared to STRs, since all maternal relatives share identical mtDNA sequences. Requires careful interpretation in forensic and anthropological studies. 7. Y chromosome analysis. The Y chromosome is inherited paternally and does not undergo recombination along most of its length. This makes it ideal for tracing paternal lineages and identifying male contributors in mixed DNA samples, e.g., sexual assault cases. Y-STR analysis focuses on polymorphic STRs located on the Y chromosome. Results are compared to Y haplotype databases to establish lineage or exclude individuals. 8. Randomly amplified polymorphic DNA, RAPD, analysis. RAPD uses short, arbitrary primers in PCR to amplify random segments of genomic DNA. The resulting pattern of amplified fragments serves as a genetic fingerprint. While RAPD is less reproducible than other methods and has largely been replaced in forensics by STR typing, it remains useful in certain genetic diversity and phylogenetic studies. 9. Introduction to recombinant DNA technology. Recombinant DNA technology is the manipulation and combination of DNA from different sources to create molecules that would not naturally occur. This technology is the backbone of genetic engineering, enabling applications such as production of therapeutic proteins, e.g., insulin, growth hormone, development of genetically modified organisms, GMOs, creation of model organisms for research, gene therapy approaches to treat genetic diseases. Basic steps in recombinant DNA work. Isolation of target DNA. Cutting DNA with restriction enzymes. Ligation into a vector. Introduction into a host organism. Selection and screening of recombinant clones. 10. Applications of DNA typing and recombinant DNA technology. Forensics, identifying suspects, exonerating the innocent, and linking evidence to crime scenes. Paternity and kinship testing, establishing biological relationships. Anthropology and archaeology, tracing human migration patterns. Medical diagnostics, detecting genetic diseases and predispositions. Agricultural biotechnology, developing disease-resistant and high-yield crops. Now that we have covered the fundamentals of DNA typing, the variety of analytical methods available, and the principles of recombinant DNA technology, we are ready to advance into the world of proteomics, the large-scale study of proteins, and explore how these tools integrate with genomic data to deepen our understanding of biology and open new avenues for scientific discovery.